So you ready for the last one? We are. The sheets that I handed out for checkout, I just need to have you have that done before you leave. Okay, it doesn't have to be done today. I just put them out there so that you would have those available and kind of know what the expectations are. We might have a few minutes at the end of the period today, but it's just your responsibility to do that before you leave. Okay? Did you get the email I sent out indicating I added a few slides on the monoclonal antibody PowerPoint, and I adjusted the lecture notes to reflect those slides were in there. So it doesn't affect anything we've already previously covered in those. I haven't changed the study guide questions either, so they're the same. Come on. Pump it up here. So what do we got going up here? What's this? It's a strep test, right? It's a rapid strep test. This is really, really nice for physicians. They can have it in their office. Basically, all they have to do is have a swab and then use that swab to inoculate these little devices right here. And within, a few minutes, you can get a reading on whether the strep is positive or negative. And this one down here with this red line that's developed is a positive strep test. Okay? Why can these be so fast? Why can they be so sensitive? What's kind of the heart of these rapid strep tests or rapid pregnancy tests? What makes them all work? There are some enzymatic reactions involved in the color reactions that develop, absolutely. But these particular tests and some of the therapies that we're going to talk about all have one thing in common, and that is that they are monoclonal antibody based. Okay, so this is an example of monoclonal antibody testing. Monoclonal antibodies are pretty cool. And they have a variety of uses, both in medical, academic, and commercial enterprises. A lot of products that you can buy over the counter or that doctors can use to detect small amounts of drugs, small amounts of toxins, small amounts of hormones, like the HCG hormone, the pregnancy hormone. We believe that monoclonal antibodies may be the key to treating certain viral diseases which have been traditionally considered to be untreatable. Okay, for example, herpes virus. Once you have a herpes virus, past wisdom has always been that this is an untreatable disease. You are stuck with it for the rest of your life. There's now some hope for a cure for diseases like herpes or maybe hepatitis, which are monoclonal antibody based. So that's pretty powerful. From an academic point of view, monoclonal antibodies can actually be used to identify and trace particular cells in the body. And that can be a really cool tool when you're a developmental molecular biologist and you want to see what does this particular cell give rise to. Monoclonal antibodies are an emerging tool in the treatment of organ rejection. Remember that I said that the primary way that organ rejection was minimized in the past was steroid administration. What's wrong with steroid administration?
Doesn't it suppress the immune system?
It generally oppresses the immune system, and the part of the immune system that we really want to suppress specifically is the T cytotoxic cell part. Okay? So can monoclonal antibodies specifically target those T cytotoxic cells and leave the rest of the immune system intact? And the answer is yeah. And then very exciting, there are monoclonal antibody-based methods for both detecting and treating cancers. And it's not limited to cancers. There are a number of inflammatory diseases which are pretty nasty, which we are now developing monoclonal antibody-based therapies. Any of you ever heard of inflammatory bowel disease before, or Crohn's disease, Some of those bowel diseases are really, really nasty diseases. They're very debilitating, they're very hard to treat, and now we're having some success with the development of monoclonal antibody therapies. So, what is a monoclonal antibody? Pretty busy slide. It's an antibody that is monospecific, meaning it reacts with only a single epitope. Populations of monoclonal antibodies are all derived from the same single ancestral cell. Okay? So if a specific B cell, for example, gets stimulated by a particular epitope, remember that that then activates the cell to go through that clonal expansion process where it divides, and we form a whole clone of cells that are identical to the original cell that was stimulated. And they will all produce a single kind of monospecific antibody, which binds to a specific epitope. That's a very powerful tool in some of those therapies and other applications we just talked about. A molecule that can target a single epitope on another molecule. That's pretty powerful. So where do we get monoclonal antibodies? Well, that's a little bit more of a problematic issue. It took a while for this particular technology to develop. So, let's say we want to develop a monoclonal antibody so we can develop a rapid strep test for Streptococcus A. Okay? So, if I take strep A bacteria and inject that into a mouse, will that elicit an immune response? Yep. And a lot of those immune cells that have been stimulated end up in places like the spleen and the lymph nodes. So could we potentially harvest nodes or spleen or something and get B lymphocytes that have been stimulated by this strep bacteria? Yeah, we could. And the spleen is a pretty easy organ to just dissect out. And so in the development of monoclonal antibodies, it's very common to challenge something like a mouse with an antigen and then give it time to develop an immune response against that, and then harvest the spleen, and then isolate the lymphocytes from the spleen. Okay? But there's a problem here. If we inject just a whole strep cell, it's got all kinds of foreign antigens on its surface. And each antigen will stimulate a different B lymphocyte. So we would get a polyclonal response. We'd have a whole bunch of activated B lymphocytes in the spleen in response to that exposure, not just one. Okay? And even if we isolated a particular molecule from the strep, let's say we had some kind of, flagellar molecule or something like that. If we injected that into the mouse, would we get a monoclonal response? Probably not, because remember, complex molecules have a number of different epitopes, a number of different antigenic determinants. And so those would stimulate a bunch of different B cells, and we'd still get a polyclonal response. Okay? So, if we have this polyclonal response, and then we isolate the spleen, we've got all these different kinds of B lymphocytes. We want only one kind of B lymphocyte, which produces a single antibody. Well, could we somehow separate out those different types of lymphocytes from one another? And the answer is yes. There are actually technologies that allow that to happen. Cells can be sorted from one another in a mixed population by various techniques, including flow cytometry. So basically, we separate the lymphocytes based upon different cell surface molecules. So, if we could get a single B lymphocyte separated out from all the rest of them, a unique type, it would produce a unique type of antibody, right? So, how about if we then do that and then pop that into some type of culture media and then let it multiply, and then all the offspring will be producing this monoclonal antibody, and then we just have to isolate that antibody that they're producing. Is that reasonable? Okay. So we inject something, get a polyclonal response, we isolate the spleen, and then use these cell sorting techniques to kind of put the individual B lymphocytes that have been activated into different wells in a culture media, and then let them multiply, and then each will produce their own unique antibody. Problem. Lymphocytes really don't grow well in tissue culture. So a great idea, but practically, this really doesn't work. Okay? Well, actually, there is a lymphocyte that will grow in culture media. That lymphocyte is called a myeloma. What kind of a lymphocyte is a myeloma? It's a cancerous lymphocyte. Okay? Remember, cancer cells have the ability to kind of grow many times when other cells, normal cells, can't. But myelomas don't produce antibody. So we couldn't somehow induce one of these lymphocytes to be cancerous so they could grow in culture and then isolate monoclonal antibody from them, because now they don't produce antibody. Or if they produce it, it's defective sometimes. So they either don't produce or it's defective. So that doesn't help us. Well, something interesting happens if you take myeloma cells and individual B lymphocytes and mix them together in the same environment. If you add polyethylene glycol, there's also a virus that you can add that will do the same thing. What that causes these two cells to do is to fuse. And when a myeloma cell fuses with a lymphocyte, it produces a hybrid cell called a hybridoma. Hybridomas will grow in culture, and they will produce antibody, and the antibody they produce is determined by the lymphocyte that the myeloma cell fused with. So every different hybridoma will produce its own unique antibody. So if we can then separate these hybridomas from one another and figure out which one of those is producing the desired monoclonal antibody, then we can just grow that cell and it will multiply and multiply and multiply and turn out a whole batch of this monoclonal antibody. Okay, so here's the kind of flow diagram up here. Here's our mouse. It's injected with an antigen. And we have then the development of a polyclonal response. Several different B sites are activated because those antigens that are injected have multiple epitopes, so we get this polyclonal response. We then take these cells and mix them with myeloma cells growing in culture. And we add some polyethylene glycol or some Sendai virus And that causes them to fuse, to form hybridomas. And each of those hybridomas will then secrete its own unique monoclonal antibody.
Is that different than the antibody that would be secreted by the...
Nope, the antibody that's produced by the hybridoma is the same one that the B cell would produce. Yep. So basically now we just need to be able to separate out these hybridomas and identify and screen which one of those are producing the desired antibody. And that has some challenges. So that's kind of the general idea. So this will get into a little bit more detail. Okay? So here's our mouse. We inject it with some kind of antigen to try to stimulate the immune response. Oftentimes you have to do multiple injections, and it takes maybe a couple of weeks for a sufficient response to develop. And in the meantime, we've got these myeloma cells growing in culture, these cancerous lymphocytes, B lymphocytes, that grow like crazy in culture but don't produce antibody. Okay, so then we harvest the spleen from the mouse. We grab some myeloma cells in culture and we mix those together and we get fusion of the cells. We get this hybridoma. So now it's got the best of both worlds. It produces antibody, but it grows in culture really well. Well, one of the things we want to do is to be able to, in this process, you've got to be able to select for hybridomas, because when you try to do this fusion, there are some various possibilities that can occur. We want this one right here. We want spleen antibody-producing cells fusing with myeloma cells. Okay? But could two myeloma cells fuse? Yeah. Could two B lymphocytes fuse? Yes. Could there be potentially no fusion at all? Yes. So what we want to do is somehow figure out a way in which we can select for cells that have done this and ignore cells that have done this or this. Okay? Well, the key to this selection of hybridomas is this particular enzyme, HGPRT, hypoxanthine guanine phosphoribosyl transferase. You don't have to remember all those names. But basically it's involved in the synthesis of nucleotides from this amino acid, hypoxanthine. Okay. Myeloma cells lack this enzyme. They are HGPRT minus. B cells have it, so they're plus. So once we've generated these hybridomas, once we've done this fusion, we throw everything onto this plate of media called HAT media, hypoxanthine aminoterine thymine. And the key is that this can only support the growth of HGPRT plus cells. So if myelomas fused with other myelomas, since they're both negative, they won't grow. B cells fused with other B cells die because B cells don't have the capacity to keep dividing the media like cancer cells do. So really, the hybridomas survive on this media because the B cell provides the HGPRT enzyme, and the myeloma provide the kind of invincibility, the ability to just keep on going and going and going. So that HAT media is a really important part of the selection of hybridomas. Okay, so they're the only ones that will grow on that particular medium.
Has they thought of using this in some kind of cancer therapy, if it can also use similar
Yeah, we'll talk about some cancer therapy. Not these cells, but the antibodies that they produce.
Right. I meant like the HAT media, if it's killing off these cancer cells. Or is it just not allowing you to?
Well, I mean, the HAT is an environment, and so how would you create an environment in the cell that way? That can be a problem with that. Okay, you think about that. Okay, so we take surviving cells from this HAT media, and we now sort them into individual wells, basically, okay, using those cell sorting techniques. But what we need to do then is to do some tests on each one of these to see, are they producing monoclonal antibodies? And are they producing the correct monoclonal antibody? Because this mixture right here of cells that we generated is heterogeneous. It's all kinds of different B lymphocytes that were stimulated by that one antigen. And we want only one of those kind of lymphocytes that produces a particular antibody. So we need to screen these hybridomas now to see which one is producing the desired monoclonal antibody. And to do that, you sample the culture media that they're growing in, because these cells secrete antibody, and you can analyze the antibodies that are produced by gel electrophoresis and Western blotting. And basically then you can use probes on that Western blot, and the probe is the epitope of the particular antigen you're looking for. And once you identify which one of those wells has the proper cell that produces the proper monoclonal antibody, then you can just grow these things in culture forever, and they just keep cranking out this one specific molecule. So this is kind of the overall idea here. Basically, you put those lymphocytes in with myelomas, select for the hybridomas, identify which hybridoma you're looking for, and then allow that to produce your purified monoclonal antibody. If you're interested, there's a link here that has a little video that shows you just what I talked about and shows you an animation of how monoclonal antibodies are produced. Well, what's really cool, John?
Is this a relatively cheap way to treat people, or is it rather expensive?
It's rather expensive right now because just pharmaceutical research and development is so incredibly expensive. And the testing to get this into clinical trials is expensive. But it's going to become more of the rule. Okay. I think really the monoclonal antibody therapy will eventually entirely replace chemotherapy and radiation probably, because this gives you a targeted way to treat tumors specifically, and you don't have all those devastating effects of those chemotherapeutic drugs and radiation stuff wiping out all the healthy tissue. So this is the way of the future goes. Yeah, we'll show you a couple of examples of drugs that are monoclonal antibody based in a second. So the idea here is pretty simple in terms of treating cancer. Okay? Here's a cancerous cell, and it expresses some abnormal cell surface molecules. And normally we would think the immune system would recognize those and eliminate them, but in some cases the cancer, that just doesn't happen. Okay? But if we can have a monoclonal antibody that we've developed against this particular tumor, and what's cool about this, think about this, you could go into the doctor if you have cancer, they can do a biopsy. And they can isolate they can take out some of your tumor cells. And that's what they could inject in the mouse to then generate the B lymphocytes that would be specific for that tumor. Okay? So now we've got this basically guided missile that will hone in on that tumor specifically. And if we could load up a payload of some kind, like a toxic molecule, onto that monoclonal antibody, then that would deliver that specifically to that tumor cell. Do you remember the name Paul Ehrlich that I referred to when we talked about chemotherapy? This was a guy that was trying to develop the magic bullet that could kill syphilis. Okay? Well, this is truly the magic bullet. Okay? This is like the laser-guided missile fired by a drone, which is very highly specific and can take out a very small target, as opposed to the carpet bombing that they would do in World War II, where they would just dump ammunition on a town and hope you got what you wanted to get. That's kind of the way chemotherapy is. It's kind of like carpet bombing. Okay? So a very specific way to kill these cells. Now, we do have some issues with monoclonal antibody therapy. Most of the original monoclonal antibodies that were produced in mice. Okay? So we challenged mice with antigen and then isolated monoclonal antibodies from mice, murine monoclonal antibodies. Okay? If we inject those into humans, what might happen? yeah, we might get an immune response against this protein. We don't want that to happen. Some later work was done to try to solve this problem, and some kinds of monoclonal antibodies were produced called chimeric monoclonal antibodies. And these are kind of part mouse, part human. And we really don't have time to get into the technology of how this is done, but they have been able to produce these chimeric monoclonal antibodies. But what they've really been able to do recently by developing transgenic mice, which can actually develop their own human antibodies, is to produce fully humanized monoclonal antibodies. And then you're not going to get much of an immune response against those. So this has been an evolution in technology, and really we have to get to this point if this is going to be a viable therapy, okay, to be using fully humanized monoclonal antibodies in therapy. It's just too risky to use murine monoclonal antibodies, or even the chimeric ones.
This summer when I was at Lilly, they were talking about monoclonal antibodies, and they'd say they would try to load the medicine in between disulfide bonds, in between, so you could get more medicine exposed to that cell other than just the one molecule.
Yeah, and they're also doing things to try to ensure that when that monoclonal antibody binds to the receptor, that endocytosis occurs, and so that drug is pulled into the cytoplasm of the cell. So there's all kinds of interesting research that's being done in anticipation of having treatment. So what's going to happen to a cancer cell if a monoclonal antibody attaches to it? Well, there's a lot of possibilities. One of the things is that this monoclonal antibody might just make the cancer cell more visible to the immune system. It's kind of like an opsonate, really, okay? And it might enhance phagocytosis. Functionally, that kick me sign, basically, that I talked about, okay? Cancer cells, to support their dramatic growth, really rely on a number of growth factors. And oftentimes the mutations that result in cells being cancerous are growth factor mutations. Monoclonal antibodies could potentially block some of those growth factors and therefore prevent the cancer cells from continuing to divide. A lot of times cancer cells are cancerous because they make extra copies of growth factor receptors. Okay? They have too many receptors, and therefore they get stimulated too much to grow. So if you can block some of those extra receptors, that can control growth. That's kind of cool. Monoclonal antibodies potentially could cause new blood cells from forming. Even from, oh, you guys didn't see that one. Have you ever seen the Cancer Warrior video? Has Morgan McComb showed that to you? Angiogenesis, cancer cells recruit new blood vessels. Okay. Monoclonal antibodies are being targeted to try to stop those new blood vessels from developing that would then effectively starve out the tumor. Delivering radiation to cancer cells, delivering chemotherapy to cancer cells, but again, in a very targeted fashion. Right now, there's a number of monoclonal antibody drugs that are available to treat various cancers, and these drugs are in all different stages right now of clinical trials. I know there are a number of them that are in stage 3 testing right now. There are some that are just being developed that are just in stage 1 testing. But again, you know that that FDA testing process takes a long time to go from conception to approval. And so these things are not particularly common right now, but they're becoming more common. Okay? And again, I don't expect you to memorize any of this stuff, but here are some of the anti-inflammatory monoclonal antibody drugs. And here are some of the applications that they treat. So you can see many of these anti-inflammatory ones treat things like arthritis, Crohn's disease, ulcerative colitis, transplant rejection, allergic reactions, asthma. Okay? And then this shows you the mechanism or target. This shows you what monoclonal antibodies really are inhibiting in many cases, okay? So tumor necrosis factor, or interleukin-2, or immunoglobulin E. You see all of these drugs names end in MAB, okay? That is indicating that they're a monoclonal antibody therapy. Oops. Here are some anti-cancer drugs, and again, names of those particular drugs and their target, and then the types of cancers: B-cell leukemia, non-Hodgkin's lymphoma, breast cancer. This one is Herceptin. You might have heard of Herceptin before. That's a pretty widely used drug now that's monoclonal antibody based to treat breast cancer. Squamous cell carcinomas. And then there are some kind of other drugs. There's one for a viral infection, RSV. And then there's one that is intended to target preventing coagulation in coronary angioplasty. So a lot of different possibilities for these very specific monoclonal antibody drugs. And this is just another list of some of the common ones. So cancer drugs, here's Herceptin, treats breast cancer. I know they've been advertising, Remicade, I know, is being advertised on TV right now to treat Crohn's disease. I know that there's an arthritis drug that's being advertised and has been advertised on TV. I don't see it on this particular list.
These are
This
ones
one.
that are already being used.
Yeah, well, these are ones that are being used at least in some stage of clinical trial or whatever. Okay, I think the one for Crohn's disease is approved for kind of general use. I know Herceptin is. Okay, Herceptin has gone through the FDA approval right now. I'm trying to think of the one that is being used to treat rheumatoid arthritis. That one's gone through the full approval process now. So this is really going to become kind of the wave of the future. I don't know if some of you have heard any rumors or not, but there's a biotherapeutic enablement company. That's what they're calling themselves. It's a group of, it's headed by a retired Eli Lilly scientist. And basically they're going to develop molecular tools, including monoclonal antibodies, that other people can use in clinical research and clinical trials for therapies. And they've primarily been a consulting company up to this point, and so they kind of provided the advice and then gotten their tools, their monoclonal antibodies from other sources. But they want to take the next step now and start producing their own therapeutic tools, including monoclonal antibodies. So we're working with them, and they want to have a partnership with Franklin College. One of the things that would happen in the partnership is that their scientists would come and teach an advanced molecular biotechnology course, things that are beyond the scope that Gordon Holmes and I could do. And they're also hoping to locate their lab facility in Franklin. So they would like to do internships then for the students, and then we could go to their lab facilities to use some of the most up-to-date scientific equipment and things. And so this could be really pretty exciting. We're kind of negotiating with the mayor and the Economic Development Commission right now to try to help them make this move to Franklin. So that could be really exciting. It could really provide some interesting opportunities for students, paid summer internships, faculty internships. It could open up a whole new world of exposure for you guys, for you probably, but for your future students, to really get into this kind of fusion between undergraduate education and applied learning in a really business, molecular research kind of environment. So we're keeping our fingers crossed about that. Okay, that's it for me. If you want to spend a little bit of time working on organizing your drawer, that would be fine. If you need additional supplies, the first place you should probably look are in these drawers over here. These two sets of drawers probably contain what you would need over here. It looks like like, Sharpies and things for your slides, or matches, or lens paper, bibulous paper, impression slides. It's pretty much all over here. So I would like to get the drawers organized. If you have things that are duplicates or extras, why don't you put them up on the front table up there? And then if you're looking for something, that might be the first place you look, because things have gotten mixed around between drawers. So as long as you can have this completed, clean up your microscope and all that stuff. And then at the very latest, turn this in to me when you do the final, okay? In terms of due dates for things, I need your urine by end of day today. You can just leave them up front up there. And then it's just a matter of your unknown by end of day on Tuesday, final Tuesday morning. If you have requests for information about flagellar arrangement, if you need that, or if there are other things, I know some of you needed the arginine test results to do your keying, and we haven't done that. You can either leave me a note up front or send me an email, and then I can check that out and respond to you.
What is food in here doing today or tomorrow?
Oh, today's Thursday. Tomorrow, end of day. Sorry. I was thinking the day was Friday.
It's the last day of class.
Last day of class. Okay.